is Dr. Aniruddha. Sorry, Saurabh, we're just been running out of time. Thank you for that wonderful presentation.
Thank you to AI Scientific Committee.
Nice not seeing you again with all that cap and mask.
Uh, I'm sitting in my clinic, so
Yeah.
thought.
Sorry, yeah. Hi, Anirudh.
Hi,
Hello,
Anil.
good morning. Good afternoon. So,
For us, yeah. Good morning to you.
yes, very good afternoon, and thank you to AIOS for this opportunity. And without much ado, I'll just start off with my presentation. So, my name is Anirudh Agarwal, and I'm at Cleveland Clinic, Abu Dhabi. And I'm just going to talk to you a little bit about what I'm doing in retinal imaging so far. And I have divided my research in retinal imaging into two aspects. One is differentiating and diagnosing retinal diseases, and quantification of retinal pathology. And what we're trying to do is using simple imaging as well as complex devices such as OCT and OCTA to get our answers in the clinic. So the first aspect that we were working on were images of choroiditis and uveitis, where we quantified the lesions into various grades 1, 2, and 3, depending on the severity of the disease. And we realized that those lesions which have grade 3 opacity, which are very yellow in color, have a high chances of recurrence and development of paradoxical worsening. This was published very recently in American Journal of Ophthalmology as clinical and imaging factors with TB choroiditis. And subsequently, what we have done is we have used artificial intelligence sort of platform where we are trying to identify these lesions automatically without sort of manually segmenting them on fundus photographs. And the aim is to automate and develop newer metrics that can help in identify these lesions which are at risk of recurrence and paradoxical worsening. So we try to make inflammations not so intimidating. And the way it works is the short clip which shows you that you just use easily available third-party softwares which can process these images, and you can run actually a very simple macros which analyze fundus photographs, and within a fraction of a second they can give you the quantification that you need, can give you the area, can mark out the lesion, and you can accordingly feed this into a convoluted neural network. The second aspect and the final aspect I'll be dealing with is OCT to differentiate and diagnose difficult entities. We know that we have lab support nowadays, which is very good. We have PCRs, we have biopsies and advanced staining techniques, but can we do something clinically? Can we use OCT to differentiate between conditions such as toxoplasmosis, retinitis, and lymphoma? So we've been working on certain key OCT features which are diagnostic. For example, you have clots of cells along posterior hyaloid in toxoplasmosis, and you have retrohyaloid hyperreflective spots in toxo, and you have these stalagmite-like deposits in toxoplasmosis. On the other hand, the involvement of only the retina as well as the choroid is a feature of toxoplasmosis, but not so in viral retinitis. So all of these findings are highly specific for toxoplasmosis. And we are trying to develop systems to add these into artificial intelligence protocols so you can diagnose these conditions with a reasonable certainty using artificial intelligence image algorithms. So part of this we have already published in British Journal of Ophthalmology, where we are comparing the OCT features in different etiologies. With our collaboration so far, we have achieved a number of papers in OCTA by using our study group. We also have formed the Collaborative Ocular Tuberculosis Study Group and also the PGI Lymphoma Study Group. So thank you very much to AIOS for this opportunity, and I'll be happy to end on time. Thank you.
Thank you, Anirudh, for taking time out and presenting this all the way from Cleveland, from Abu Dhabi.